if there is anything else that you want me to repeat, cover, or whatever, what I'm trying to do is that get somebody online or in class to give you another purview of what is going on. Okay? I'm not saying it, but pretty much if you have attended all these 20 lectures, you still have all the maximum points you already have. Okay? And if you missed it, so we'll compensate for that. If you missed like 2 and you attend next 2, we'll compensate those points. Okay? So 20th is the last of all the series of immunology, and it covers the most important thing, the diagnostic tests. So you'll come across many diagnostic tests in your practice. So I'm going to give you theoretical basis for that, or immunological basis for that. And again, keep that in mind that if you remember the first few lectures, antigen-antibody complex. All these things, all the tests are based upon antigen-antibody complex, right? To as simple tests as pregnancy test, or the sore throat test, which I told you, the step test, or any other test which are available, immunological based diagnostic tests. So this will be the basis. And I'll just give you a synonym or a concept. In business they say, if you have money, we have a product. This is a business kind of slogan. So the tests are based upon, if you give me an antigen, I have an antibody for you. If you give me an antibody, I have an antigen. So antigen-antibody is the test, right? So we take samples from the patient, right? Whether it be blood, serum, saliva, vaginal, fluteral, any fluid. And either we look for an antigen or we look for an antibody. And this antigen-antibody complex will give you the result. So this is the basis for that. Okay? Now this one shows you one of the tests that is there, which we call immunoassay. Immunoassay, as the word suggests, is a test that employs the properties of many proteins to adhere to plastic and form a molecule or a layer. Antigen is applied to plastic wells. Antibodies are added. The cell is washed, and antibodies bound antigen are made by the use of radiolabel or enzyme like immunoassay. So we usually call it enzyme-linked immunoassays. Enzyme-linked immunoassays. But keep that in mind that I told you the antigen-antibody will combine, but how do we see that? So in order for us to see the color, right, we have to do something. So what happened is that in this case, we label the antigens, right? And then antibodies are less as compared to antigen, and then we give them a chance to bind with each other. So of course, antibodies have this antigen binding sites, like Fab, and they will bind with this antigen. You can see from here, they are binding with antigen. So in this case, most of the time, there are 2 arms, so I can hold 2 things. So one antibody can bind to 2 antigens. So you can see from here, once they are bound, so we have some free antigen, but then we separate this bound one with the rest of them, and then you can see from here, in this case, we started up 1, 2, 3, 4, 5, 6, 7, 8, 9, and then we had 1, 2, 3, 4, 5, 6 of them got labeled over here, and 3 were redundant, so we had 6 units, right? And 3 were not able to bind. So this way we can kind of tell, the concentration or the titer of the antigen or antibody. Okay, and these, since they are proteins, so you can see amount of label bound to antibody after incubation for constant amount of antibody and label antigen. Did anybody ever see the well plate, 96 well plate? Have you seen that? 24 well plate? Okay, maybe I'll someday, maybe next week, I'll take you to the lab. If I don't get any of the visiting lecturers, so I'm just going to take you to the lab and actually show you. Okay, and I did see there's one lab as well, but we'll just walk you through the lab and give you the points for just being there. But at least we'll give you a plate. So in the plastic plate, we put the antigen and pour on top of that antibodies in the mix. So this is one way of looking at the tests, right? But the thing is that once we know how many antibodies we enter, and we can label the antigen, and these antigens will be then figured out in terms of what were labeled versus unlabeled. So that's a simple test. Okay. The other test that we do in the second case is that in this case it is a radioimmunoassay. So this was a simple immunoassay. We call it solid phase immunoassay, right? As compared to so this is first test. The second test is radioimmunoassay, right? So you'll see many tests are there, and this case got radioimmunoassay. Again, we have antigens, we have antibody. We're going to mix them together and find out, if they are radiolabeled, because radiolabel will shine. Radiolabel is like it's going to give you a color. Okay. In this case, we have labeled and unlabeled antigens. Some of them are labeled and some of them are unlabeled. So you have, say 9 units in each. So let's say 1, 2, 3, 4, 5, 6, 7, 8, 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 are labeled and 9 are unlabeled. So we pretty much know. You didn't know they were 9, you're laughing, right? You just drew it. You are lucky that you drew 9. I can guess by your face. Then what happened is, then we have the addition of this antigen specific antibody. These antigen which are specific for this antibody, we put it over there, and then they are going to bind. They are going to bind. And then we can separate them based upon their binding, whichever will bind. Okay, because keep that in mind, the most specific reaction in the body is antigen plus antibody, right? Which is antigen-antibody complex. So that's like a binding, very specific, based upon the genes. Okay. Now what happens is that this is more of a radio label, which is give you like we have this labeled versus unlabeled with this radio label, and there is a competition of the non-labeled and labeled antigen, right? Because there's going to be competition between who's going to label, and this will give you an idea. Like for example, in this case, bound labels 3 units. Right, and 6 units unbound label and 6 units. So we can go the quantity. You can determine the quantity of an assay in terms of the ratio that we may have. Like in this case, it is 3 to 6, which were bound, which were unbound. So this is how like a radio assays we do in terms of concentration. Because keep that in mind that we take, like for example, an antigen of COVID from the patient. You see those COVID things, and it has a labeled antibody. So they put over there, and they will be bound together and give you a color. So that is this enzyme they add to give you the color. But this one is more specific. It's a radio label, right? So radio label means isotope. That wouldn't be done in the home kit. That's a special one. Because you want to give radio label assay, but it's more specific, right? So they're going to make this COVID-19 test and give it to anybody, and you can throw it or whatever you want to do. The only thing you have to take care is the biological of this, what you call the antigen sample. Other than that, you put the gloves. But radio label is serious because that's done in a machine. Like for example, you want to do the quantity of thyroid hormone. the quantity of the other hormones, so they will be labeled. And they usually are radioisotope labeled. So that test is done, but you have to keep in mind that's based upon the principle of antigen-antibody binding. Any questions on that? Right? So we have an idea as to what the concentration is of a protein. Okay. More often in the clinical practice, it's called ELISA. ELISA. Okay. What is the the Amazon walkie? What is it called? You know,
Alexa.
they have
Alexa.
Alexa. So that's not, it's not Alexa, but it's ELISA. ELISA, enzyme linked immunosorbent assay. It's not a Z, but a S, as SAN. So it is a solid-based immunoassay, again immunoassay, which an enzyme is linked to the anti-immunoglobulin antibody, and then quantification is achieved by colorimetric evaluations. You see colorimetric spectrophotometry, if you see, you know, the old biochemistry experiment, you just add a color, which an enzyme that gives you color. And you can see from here, after the addition of a substrate which changes color in the action of the enzyme. So that's the principle, biochemical principle. It changes color. So you add the substrate which changes the color of that particular enzyme, if that's a role. It's called. So what you normally do is, for example, again some of the tests, strip tests, they call strips. You see pregnancy tests and, you know, other infection tests. So you will see they say put the drop of urine over there, okay, or put a sample of sputum over there, or just take a throat swab. So sampling can be different depending upon different sites. Okay. So what happens is that we have this again a plate, right? I told you like a small plate, and you can actually Google it as well. Just Google 96 well plate, you'll see what I'm talking about. This is small little plate, like 6 inches by 3 inches or 9 inches. So it has different wells. One is called 8 well plate, other is 24 well plate, and the one, if you put like 96 well plate, you will see that little like wells over there, which will take about like 300 microliter of the fluid each time. Okay? So what we do is that, first of all, we take this plate, take the sample which has an antigen, and then we coat this antigen coated plastic well. So this has an ability, right? Ability to bind to the plate. Okay? And then what we do is, after that, actually we do it before that, we block unbound sites on the plastic with unrelated protein. So for example, you know, if there are sites over here which remain unblocked, so we add a blocking protein, you can see a blocking protein, and that kind of fills in the gap, right? So there's no more room for any antigens. Because antigens can be proteins to actually block it. Okay? So you have a plate, and you add the antigen on top of that, and the antigen binds to the bottom of the plate, okay, because they are molecules. And then you find out the sites which are available to fill in the unbound spaces like protein. And then what you do is, then you add a enzyme labeled antibody. So you have an antibody, again the antibody, you know, and this has this fab, 2 fabs, right? And this is FC portion. And then you add the enzyme over here. So since they are antigens, all of them are antigens, so they will bind with fabs, and their top position FC portion, you know, will kind of jut out because they're going to bind to it. So this is one step. So what has happened is that, because this is a specific, we have to rule, the principle is that antigen and antibodies are lock and key. So if you have the right key to the right lock, it's going to open up. So if it is right, sample, right thing. So what will happen is it will bind, and we now want to find out that which one of them has really bound, right? So what will happen is that when we will add a substrate. So you can see the substrate is added over here in this situation. So substrate will, as the principle says, when you add a substrate, the action of the enzyme will be obvious. So we'll see this one will shine. So this one over here will shine, change the color. So you'll put over there, if for example you see in pregnancy test or steptoe test, you will see you put the sample and after a while you see the color changes. So that will show positive, and they have control as well. So they have the control to test whether they'll fall. The only thing you have to keep in mind about false positive, false negative, and then you don't have to worry at this level of the course, but they are important that the tests are very specific, right? And they are very sensitive. Specific means specific for that particular antigen. Sensitive means that they can pick up even a small amount of antigen or antibody. So far so good. So solid phase amino acid number 1, radio amino acid number 2, and ELISA number 3. So this is the third test, right, where we code the antigen and put antibody on top of that. Okay. We can technically also reverse as well. What we can do is that instead of coding an antigen, we can also code with the antibody. Right? And binds over here, and then you put antigen on top. You can do that. But this is the better way of doing it. But as I said, if you have an antigen antibody, either you can pick up like antibodies in the serum or blood, or you can put antigens for that. Okay. All right. The other one over here is another very important tech. We have that machine over there in our lab. I'll show it to you next week. Right? It is called FACS. Very common test. It says fluorescein activated cell sorter. That's what it stands for. So this is number 4. Fluorescein activated cell sorter, right? Is a method in which antigen is detected by the use of a fluorescence labeled immunoglobulin, right? In direct immunofluorescence, the antibody to the antigen in question carries the fluorescent label. That's like direct. In indirect, the immunofluorescence, the antigen-specific antibody is not labeled. It is detected by the addition of another antibody. So we can quantitate number of cells over there. So in this case, we usually look for either for these tests, we can look for antigen concentration or antibody. Like for example, if you have infection, you may have an antigen for bacteria, antigen for virus, antigen for parasite, right? That's one way, immediately. But your body will kick in to form antibodies. That may take a while. So immediate test will be an antigen based, but later test could be an antibody based. Okay, and both of them are in your body. But the other test is that then you look at cells, because I told you that some of the cells, whether T cells and B cells, they proliferate, they expand, right? So we can either go for looking for an antibodies, or we can also go for looking for cells. So what we do is that, for example, we'll take your blood out, and then we will prepare the cells. We'll just, like in this case, let me give an example. So we take the blood, and on the blood we take PBMCs, polymorphonuclear cells, right? Separate them by the kits that we have, right? And we make a cell mixture suspension. So we have a suspension which has all different cells, right? And then we enter the fluorescent labeled antibody. So we have antibody for that particular cell, right? for this one, this is, for example, this cell are we looking for example, let's say this is a T cell and a B cell, and we want to find out how many T cells are there, right? And then again, we can look for T cell and B cell depending on the marker. We can also look for CD4 positive cell, CD8 positive cell, and any other cell that we can. And we can also look for B cell or any other cell because they have that tag attached to them. And we make antibody for those cells, right? And these antibodies will locate and bind to these cells, either this one or this one. You can see they actually are binding to them, right? And then we have a fluorescent antibody which is going to give a color. And then we have a machine. We have a machine over here, and which has a beam, like a very small. You can see from here. If we have this one, and then there is a very small beam. We put the cells over here, and this is so small beam that one cell at a time can pass. So cells usually are 15 to 20 micrometer, very small, right? And then there is a light source over here, like a laser light. We may have different spectrum of light depending upon what lights we are looking at. So we can detect hundreds of different blue, green, yellow, orange. They are all labeled antibodies. So this light goes over here, and there is a camera at the back, and the camera will pick up how many cells are there, right? And then this machine can also sort things out. So what it can do is that you put all the cells together, and this will put T cells, B cells, macrophages, and other cells separately. It's going to sort them out in different containers. It will also give you the number of cells. Like for example, in AIDS, you have, normally for every one CD8 cell, you have 2 CD4 T cells, so 2:1 ratio, right? Or for example, you say how many, look at how many neutrophils do I have, number of neutrophils. This is how they calculate that. They have a huge machine that calculates neutrophils, lymphocytes, B cell, T cell. You just put blood sample over there, and the machine is going to do the rest of it based upon the technology. And then it will kind of make a chart for you on a logarithm base as well, and then you can see depending upon with cell density, the size, right? It will form clusters, right? And then again, if you have labeled with an antibody over here, you can see from here, there's a detector over here, and there's a deflection plate, and laser beam will come over there, and then fluorescent cell will be separated from non-fluorescent cell, right? So for example, if you put, in this case, we put just one antibody, which have this color, so we'll have like one fluorescent cell, and rest of them will be there, but you can put more than one. These days, they put like hundreds of them. So it is a huge machine. It's going to separate. It's like an amazing thing. Very commonly used. So it will, the physicians will request like, how many T cells, how many B cells, how many CD4 T cells, so this depending upon the size of the cell, and like platelets are very small, and the macrophages are bigger. So they have, and then tagging, we call it tagging by the fluorescent antibody. And if you go and look at it, and I'll show it to you, we have that machine over here. We have like a small, but in the hospital they have industrial level of thing. It's going to give you the exact number of monocyte, lymphocyte, eosinophil, neutrophil, this percentage, this number, right? So because of this technology that they have. So this is a fluorescent activated cell sorter, FACS, very important test, and this is the principle that we have cell suspension, we have fluorescent antibodies, we have a detector, and then we can also detect and separate. It's a cell sorter as well. You can sort cell out, okay, depending upon what the label. Okay, so this is what it is, production of monoclonal antibodies. And you remember that we sometimes give monoclonal antibodies as a treatment, passive immunization, tetanus toxoid, okay? and we have to make this specific antibody. So body makes it, right? And in this case, what you will see is that we usually have a very important cell line, which is a cancer cell line. So cancer expand out of control, right? So this is like one of the cancer which is called myeloma. So we get these cancer cells from mouse myeloma, cancer cell myeloma, right, which has the ability. And then we have an antigen, whichever antigen we want to prepare, very specific antigen, right? And we kind of fuse them together. So we give this antigen and those cancer cells, right? And when we do this fusion, right? You can see there's a fusion, and then some of the cells will die. And the idea is that, you know, the cancer cells are, what is the major quality of the cancer cell? You did the DAS one, right? Oh, pathology. What is the major characteristic of a cancer cell?
Uncontrollable growth.
Yes, very good. What else? They're immortal. They're gonna kill you, but they keep on growing immortal. There's no limit to that. That's why people die. They're immortal. So these are immortal, you can see from here, immortal hybrid cell lines in this media, and then we kind of take up the clones of the antibodies that are made. Then we go clone 1, clone 2, clone 3, right? And we want to, the idea is that we just want to have an antibody for this particular antigen that we want to make. Like for example, if we want to make an antigen against tetanus, so give the tetanus antigen, so we have very specific, similar kind of antibodies, you can see. These antibodies which have from the same clone, one clone we call monoclonal. Monoclonal. In some of the drugs that you will see, you know, once they teach you, what they do is that there's a name of drug, and at the end it will say MAB. The name of the drug will say MAB, which means monoclonal antibody. So that drug, you know, if it ends with MAB, this means it is an antibody. It's called monoclonal antibody, and again it is based upon similar identical antibody that's there, and it will give all the tests that we talked about. Okay, and this is called hybridoma technology. We call it hybridoma technology, right? Which we have a cells producing, and then many cells are made for monoclonal antibody production. Okay. Then lastly, we have another. These are all the cells, and then we make the antibodies. And this one again is for cells when we are looking at cell death. In this test, we are looking for cell death, and the cell death especially for apoptosis. Apoptosis, we already discussed that, right? So what we have is that we want to find out assessment of apoptosis using tunnel assay. Just have to remember there's a test called tunnel assay, and that is to look at apoptosis. So this actually goes with this one, right? Monoclonal antibodies are highly specific agent consisting of homogeneous population. So this is again something to do with this, correct?
It was separate, number 4 and number 5, but yeah.
Yeah, but basically these are monoclonal antibody. This is a separate one. Now, because that's again describes this hybridoma technology. So we have a nucleic acid, like for example, this is the DNA, because in apoptosis there is one of the things. What is the hallmark of apoptosis? Hallmark of apoptosis, very common question asked. That's the definition of apoptosis. What is the hallmark of apoptosis? Okay.
Can it come back?
Huh?
Is it what
it's what Anita said, but the hallmark isn't that it's done, like, it's certified that it can't come back,
The hallmark of it is hallmark of it is identification, which is called DNA fragmentation. Fragmentation. So if you see in a cell DNA is fragmented, this shows apoptosis. This is called hallmark of apoptosis. Very
So
common
that's how you
question.
identify it, if you see that
Yeah.
it's fragmented.
Yeah, if the cell has a fragmented DNA, it's going to apoptosis. That's why we have to detect that. So what we add is, you can see where it says DNA. So DNA is fragmented. So we have to add a marker to find out if the DNA is fragmented. This is the DNA, is broken. So we add a fluorescent antibody, right? And then detect the DNA fragmentation. So DNA is fragmented, the cell is dying, right? So we have to label it with an antibody and test it as a cell death, right? And that's called tunnel assay. And the hallmark of apoptosis, which is a very important concept in medicine. And what happens to the apoptotic cell when it is defragmented? This is again, what does the body do with? Again, I'm going to ask you the same question, that what's the difference between inflammation and necrosis? But anyway, these are something that you need to know on the finger trip, because if you have to understand how does the cancer work. So what happens is, when a cell dies by apoptosis, it is phagocytosed by other cells like neutrophils and megaphils. So it's a clean demolition. Like you have your red blood cells, millions of them, replaced every 4 months, but you don't kind of bleed out or something. So they are worn out, die, and the other cells will phagocytose and kill them. As compared to necrosis, which this will never happen. So this is the basic, this is what the DAS1, I believe, or pathophysiology of the cell death is. You have to understand before you understand cancer, how to treat it, and so on and so forth. Okay? So this is a measurement of cell death by apoptosis using an assay where you take the DNA, look at the DNA, add the fluorescent marker over here, especially antibody, and it's going to shine and it will tell you the cell has died. So far so good? No? Okay. Any questions? Okay, so that pretty much ends the immunology course, all the way from the first lectures for antigen antibody, and then again the recorded lectures is available. This will be available as well, what I'm saying right now. And so when you look at the